Have you ever, sat down and really thought about how life began?
Like, where did it all come from?
Yeah, it's a question that people have been asking forever, and today we're going to dive into some really cool research that tries to answer that.
Yeah, buckle up. This is a good one.
We're going to be looking at a lecture by Dr. James Tour,
He's a big-time chemist at Rice University.
and he has a super interesting perspective.
What's cool about this deep dive is that Dr. Tour isn't just some guy theorizing. He's actually out there making groundbreaking discoveries in nanotechnology.
Oh, yeah, he's known for making some really wild stuff.
Like tiny nano cars that are powered by light.
And get this, he's got a method for turning everyday materials like bread into graphene.
Crazy, right?
But the most interesting thing is how he uses all this experience building stuff at the molecular level to help us understand the origin of life.
Yeah, the challenges of abiogenesis.
Exactly. And he makes this really brilliant comparison. He's like, imagine trying to build a car
From scratch.
from scratch, but with no instructions.
And no tools.
Even if you had all the parts laid out in front of you.
It would be insanely hard, wouldn't it?
Oh, absolutely. There's no way I could do that. I can barely change a tire, let alone build a whole engine.
Now imagine that, but 1,000,000 times harder.
Oh, gosh.
Because building a cell,
A cell is infinitely more complex than a car.
way more.
And here's the thing. On early Earth, you wouldn't even have all those pre-made parts like you would with a car.
You just have basic compounds.
Like what kind of compounds?
Like carbon dioxide, ammonia, nitrogen, stuff like that.
So it's not even like putting a puzzle together.
No.
It's like you have to make the puzzle pieces first before you can even start fitting them together.
Exactly. It's a whole other level.
Wow. Okay, so that's where Dr. Tour's perspective as a chemist really comes in.
He really breaks down all the intricate chemical hurdles that make the origin of life so hard to explain.
Right, so let's get into it. What are some of those hurdles?
What makes this process so hard from a chemical standpoint?
Yeah.
Well, one of the biggest challenges is this thing called chirality.
Chiral.
It's this idea that molecules can exist in two forms.
Kind of like our hands, right?
Yeah, like your left and right hand.
Mirror images of each other.
Exactly. They're both hands, but you can't superimpose them on each other.
Right, I remember learning about that in chemistry class.
So the fascinating thing is that living systems almost always use just one type of handedness for their molecules.
Like all right-handed or all left-handed?
Exactly. For instance, the sugars in DNA are all right-handed.
Okay, so how did this preference for one-handedness happen?
How did that arise in a prebiotic world?
Like without anything guiding them to choose the correct handedness?
That's a question scientists are still trying to figure out.
Makes sense.
And it gets even more complicated.
How so?
Because in a lab, chemists have all these fancy techniques to control their reactions.
And to purify them.
Right, but on early Earth, there wouldn't have been any of that control.
So are you saying that these molecules would have been constantly reacting?
Yeah, just a chaotic mix.
With no way to stop at the right point?
Or to get rid of any incorrect additions.
Yeah, that makes it way harder.
Think of it like baking a cake, but you have no control over the temperature
Oh, no.
or when to stop baking it.
So you're saying it could end up burnt?
Or it could be a gooey, undercooked mess.
Okay, yeah, I get the analogy.
See what I mean?
So we've got this chirality problem. We've got these uncontrolled reactions.
Right.
What other chemical nightmares are we dealing with?
Well, another big one is the order you add things in.
Oh, right, like in a recipe?
Exactly. In chemistry, the order you add chemicals to a reaction is super important.
It can make the difference between success and failure.
Totally. It's
Yeah.
just like following a recipe. You wouldn't add the frosting to a cake before you've even baked the batter.
Right, the order has to be precise.
And the conditions have to be just right, too.
So it's not enough to just have the right ingredients. You have to add them in the right order under the right conditions.
Exactly. It's like a delicate dance
And it sounds like a lot could go wrong in a prebiotic environment.
without some intelligent guidance.
Yeah.
In fact, Dr. Tour gives this really fascinating example from his own research.
What's that?
He was working on synthesizing a molecular motor.
A molecular motor. What's that?
It's like a tiny little machine at the nanoscale.
Oh, wow.
And it required him to control the temperature perfectly.
And to get the order of reagent addition exactly right.
Every single step had to be perfect.
So is he implying that molecules on early Earth would have needed some kind of advanced lab setup?
To achieve that level of control,
Yeah.
well, he puts it a bit more humorously.
Ow.
He's like, imagine these molecules having to travel between glaciers
Yeah, Kano's hot springs.
to get the perfect conditions for each step.
Yeah, like they're going on a road trip to find the perfect lab.
That's funny, but also kind of scary.
It really highlights how specific the environment would have to be.
for these reactions to even have a chance.
Exactly.
And even if these reactions somehow did create complex molecules.
There's still another hurdle.
What's that?
Characterization.
Characterization? Yeah, it's basically knowing exactly what molecule you've created.
So it's not enough to just make something.
Nope.
You have to analyze it and be sure it's what you intended to make.
Exactly. Scientists use really sophisticated instruments for this.
But those wouldn't have existed on early Earth.
Nope, not a chance.
Wow. So not only do you need these crazy complex reactions to happen,
Against all odds.
but you also need a way to check what you've made.
It's mind-boggling.
I'm starting to see why Dr. Tour's experience building stuff at the molecular level gives him such a unique view on this.
On how hard a biogenesis really is.
Yeah, it sounds like he has a much deeper appreciation for the complexity.
Than someone who just theorizes about it.
For sure.
And we've only just scratched the surface here.
What do you mean?
We haven't even talked about assembling all these components into something that looks like a cell.
Oh, right. We're still just talking about making the building blocks.
Yeah, this is just the beginning.
It feels like we're uncovering layer after layer of complexity.
It's a deep rabbit hole.
But that's what makes it so fascinating.
It's a good one.
So what happens when scientists try to build these hypothetical precursors to living cells?
Those so-called protocells.
Do those experiments give us any clues?
That's a great question.
Yeah.
And that's exactly what we're going to explore next.
All right, listeners, we're just getting started.
Stay tuned for part 2.
We're going to find out how those protocell experiments stack up against what a real functioning cell looks like.
It's going to be good.
Don't go anywhere. All right, so in the last part, we talked about all those insane chemical challenges to life just like popping up out of nowhere.
It's mind-blowing, isn't it?
Totally.
Yeah.
But let's just say for a minute that those early molecules somehow did beat the odds.
And they managed to make the building blocks of life.
Yeah, how did those building blocks then, like, organize themselves into something that even resembles a cell?
Well, that's where the idea of protocells comes in.
Protocell.
Yeah, they're these hypothetical precursors to living cells.
Like a stepping stone from non-living stuff to life.
Exactly.
Okay, I've heard of protocells.
They're usually shown as like these simple bubbles of lipids.
Yeah, with some basic molecules inside?
Right. But I have a feeling it's not that simple, is it?
Probably not. Dr. Tour seems pretty critical of how oversimplified this idea of protocells has become.
Oh, yeah, he really goes after that.
So what's wrong with the typical picture of a protocell? What are we missing?
Well, for starters, most experiments use just one type of lipid to make these protocells.
Okay.
But real cell membranes are incredibly diverse.
Oh, really?
Scientists have found over 40,000 different types of lipids in living organisms.
40,000 different types?
Yeah, it's crazy.
Wow. I guess it's not just any lipid that'll do the job then.
No way. And it gets even more complicated.
How so?
Real cell membranes are asymmetrical.
Asymmetric.
The inner and outer layers have different types of lipids.
Huh. Interesting. So it's not just a uniform bubble.
Nope, not at all. We haven't even figured out how to make something that complex in a lab.
Let alone imagine it happening randomly billions of years ago.
Exactly. It's a huge challenge.
So it's more like trying to build a house with walls made of brick on the outside and wood on the inside.
With each layer having a specific purpose.
Why is that asymmetry so important?
Well, different lipids do different things.
Okay.
And how they're arranged within the membrane affects things like transport and signaling.
And the overall stability of the cell?
Exactly. It's a level of sophistication we're only starting to understand.
So we're not just talking about a simple bubble here.
No way. It's super intricate.
With a specific architecture.
Absolutely.
And we haven't even talked about all the other stuff that needs to be in that membrane.
Right. All the proteins and carbohydrates.
Yeah, it's like we're adding more and more layers to this onion.
And speaking of complexity, there's this one class of molecules that often gets overlooked.
What's that?
Carbohydrates.
Carbohydrates, aren't those just sugars?
Well, yeah,
Yeah.
but they're way more important than most people realize.
Really?
The surface of a cell is covered in these complex carbohydrate structures,
Okay.
and they carry a ton of information.
Interesting.
Yeah, even more information than DNA itself.
Wait, hold on. More information than DNA.
Yeah, think of carbohydrates as the language that cells use to talk to each other
Okay.
and to interact with their environment.
I always thought DNA was the ultimate blueprint for life.
It is, but DNA is like the book, and carbohydrates are the language the book is written in.
That's a good way to put it. So how much information are we talking about here?
Well, imagine a simple sugar molecule with just 6 units.
6 units.
There are over 1 trillion ways to connect those 6 units.
1 trillion.
Yeah, it's a mind-boggling number.
I'm starting to see why you said they carry more information than DNA.
It's crazy, right?
But why does this matter for the origin of life?
Well, if those sugars aren't connected in the right way,
cell won't work.
It could even be fatal for the cell. It's like trying to write a book with a language where you can combine the letters in a trillion ways.
But only one specific combination actually makes sense.
Exactly.
Yeah.
It's a level of precision that's hard to even grasp.
So you're saying not only do you need to create these complex carbohydrates
But you also need to put them together in the exactly right way.
for the cell to actually function.
Precisely. And to put that trillion into perspective,
Yeah.
it's way more than the estimated number of particles in the entire universe.
Whoa, that really puts things in perspective.
It's a level of complexity that's almost impossible to fathom.
We're talking about numbers that are just beyond comprehension.
And even with all these complex molecules and structures, there's something even more fundamental that's missing.
What's that?
We haven't talked about the interactome yet.
The interactome, what is that?
It's the network of interactions between all the molecules in a cell.
Like how they communicate and work together?
Exactly. It's like a molecular social network.
Okay, I kind of get it.
Think of it like this. You could have all the right ingredients for a cake,
Okay.
but just mixing them together won't magically give you a cake.
You need them to interact in specific ways.
Right. The flour needs to absorb the liquid. The baking powder needs to create bubbles.
For the cake to rise and bake properly.
Exactly.
So the interactome is like the recipe that tells all the parts of the cell how to work together.
To create a functioning whole.
That makes sense.
These interactions aren't as strong as chemical bonds,
Okay.
but they're crucial for the cell to operate.
So it's all about precision and timing.
It's like an incredibly intricate dance between molecules.
It's hard to imagine that just happening spontaneously.
And that brings us to a fundamental question.
What's that?
Where did the information for all of this come from?
Ah, the information problem.
Right. It's not enough to just have the building blocks
You need the instructions.
on how to assemble them and how to make them interact in meaningful ways.
It's like trying to build a computer.
By just throwing a bunch of transistors and wires into a box
Exactly. You need the blueprint, the code, the software.
to tell those components how to work together.
So where did the information in DNA and RNA come from?
Where did the blueprints of life come from?
That's a big one.
We've barely scratched the surface of creating the building blocks of life in a lab
And we're nowhere near figuring out how to program them.
to create the software of life.
Right. That's a whole other level of complexity.
And this leads us to some of the claims that are being made about synthetic cells.
Oh, yeah. I've seen headlines about scientists creating synthetic life.
Dr. Tour has some interesting things to say about that.
Does he think they're legit?
Not really. He thinks a lot of the claims are overblown.
Really? Why is that what's misleading about them?
Well, he uses the example of Craig Venter's team.
They were the ones who made headlines for creating a synthetic cell back in 2010.
Exactly. But what they actually did was copy an existing bacterial genome
Okay.
and transplant it into another cell.
So they didn't actually build a new cell from scratch.
Nope. It's like taking the hard drive from one computer and putting it into another one.
Yeah, you're not creating anything new. You're just replicating something that already exists.
Right. And Dr. Tour is equally critical of a 2018 study that claimed to make lifelike artificial cells.
What did they do?
They used prefabricated plastic microcapsules, Clay, and purchased biological components.
So it's like saying you built a car,
By taking a plastic toy car.
filling it with sand and gluing some wheels on.
Exactly. It might look like a car,
But it's not a real car.
and it's not a functioning, self-replicating, autonomous entity.
That's a good point. It's important to distinguish between, like, mimicking some aspects of life.
And actually creating life itself.
Dr. Tour has this funny analogy to describe these protocell experiments.
What's that?
He says they're like buying 20 pounds of sliced turkey meat, adding a gallon of turkey broth, warming it up, sticking in a few feathers, and then saying you've created a live turkey.
That's hilarious.
But also kind of disturbing.
It really highlights how far we are from truly understanding and recreating life.
From scratch.
Yeah, we're not even close.
There's a lot of hype around synthetic cells.
But it seems like we still have a long way to go.
To truly grasp the complexity of life.
Absolutely. And this awareness of complexity is what leads Dr. Tour to call for a timeout in origin of life research.
A timeout that seems pretty drastic. Is he saying we should give up on this whole field of research?
Not necessarily. He's just urging the scientific community to step back and reevaluate our assumptions
To acknowledge what we don't know.
and maybe explore some new approaches.
So a more critical and reflective approach.
Exactly.
He thinks that continuing down the same path without addressing these fundamental problems.
They're not going to get us anywhere.
So he's calling for a shift in thinking.
A fresh perspective.
And this leads us to another topic that's closely related to the origin of life.
Evolution.
Evolution. That's always a fun one. I'm curious to hear how Dr. Tour approaches this given his unique perspective.
As both a scientist and a Christian?
Not exactly. Does he see them as compatible?
Well, get ready for some thought-provoking insights, because Dr. Tour doesn't shy away from tackling evolution head-on.
Okay, I'm all ears.
He digs into the mechanisms of change, the theory of universal common descent, and all the evidence for and against these ideas.
All right, listeners, we're about to enter a whole new layer of this deep dive.
It's going to get interesting.
Get ready to challenge your assumptions as we explore the complex and often debated world of evolution. Welcome back to our deep dive into the origin of life. So in the last part, we left off with Dr. Tour calling for a timeout in origin of life research, saying we got to rethink some things and maybe try some new approaches. But what about evolution? How does Dr. Tour square his science with his faith when it comes to that whole debate?
What's really interesting is that Dr. Tour doesn't shy away from evolution at all. He dives right in and makes this key distinction between the mechanisms of change and the theory of universal common descent.
Wait, I'm not sure I get that. Aren't those basically the same thing?
Not exact. Think about it this way. We can see mechanisms of change happening right now, like mutations and natural selection. But universal common descent goes further. It says that all life on Earth shares a common ancestor. It's a big statement about the whole history of life.
Okay, I'm starting to see the difference. It's like the difference between seeing how a car engine works and saying that all cars came from one single original car. One's about the mechanics and the other's about family history.
Exactly. And Dr. Tour says the genetic evidence for common descent is pretty convincing. But he also points out some challenges to this idea, things that make you stop and think.
Okay, I'm listening. What are some of those challenges?
He focuses on what he calls the uncommon parts of human DNA stuff that doesn't fit neatly into this idea of simple, gradual evolution from one ancestor.
Uncommon DNA. That sounds kind of mysterious. What's he talking about specifically?
One example is what used to be called junk DNA. That's the huge chunk of our genome that doesn't code for proteins. People used to think it was just useless leftover stuff, but now we know it's super important for regulating genes. It controls how and when genes get turned on and off.
So it's not junk after all, but how does this challenge common descent?
Well, these non-coding parts of DNA show big differences between species, even those that are considered closely related. So while the protein-coding genes might be similar between humans and chimps, the way those genes are controlled can be quite different.
Interesting. So it's not just about the genes themselves, it's about the control mechanisms too that adds another layer of complexity.
For sure, it suggests that the differences between species go way beyond simple changes in the genes that make proteins. And this leads Dr. Tour to another fascinating piece of the puzzle: orphan genes.
Orphan genes, what are those?
These are genes that are unique to just one species or a small group of organisms. They often do important things for specific traits. And the fact that they pop up seemingly out of nowhere in the evolutionary timeline is hard to explain through gradual descent.
It's like finding a brand new car part that doesn't fit any existing car. Where did it come from? So these orphan genes suggest there might be some jumps or gaps in the evolutionary tree.
That's one way to look at it. And then Dr. Tour gets to what he sees as the biggest difference between humans and everything else on Earth: our capacity for symbolism.
Symbolism? What do you mean by that?
She's talking about our unique abilities for art, music language, math, religion, this broader organization of symbolism, he calls it. These are the things that set us apart even from our closest relatives, like chimps.
Yeah, we haven't found any chimps writing symphonies or solving complex math problems. It does seem like there's a big leap, something special about human consciousness.
And Dr. Tour challenges evolutionary biologists to explain this massive and presently unexplainable infusion that gave us this capacity. If humans and chimps share a common ancestor, what happened in that short amount of evolutionary time to cause such a huge difference?
That's a good question. He seems to be pointing out a hole in our current understanding of human evolution.
And this leads into another important point: the mechanism problem in evolutionary theory.
The mechanism problem, what's that all about?
It comes down to this: how do complex biological systems actually change? He uses the example of body plans, the basic layout of an organism, like where the limbs and organs are.
Yeah, like the difference between a fish and a bird.
Exactly. And while there are ideas about how body plans evolved, the actual step-by-step mechanisms for these big changes are still a mystery.
So it's one thing to say that wings evolved, but it's another to explain exactly how that happened at the molecular level.
Right, and Dr. Tour says that any major change in a complex system would need a bunch of mutations to happen all at the same time and in the right order. While evolutionary biologists often talk about small, gradual changes, the odds of a ton of specific mutations all happening together are, well, really tiny.
So it's like winning the lottery over and over again. It's not impossible, but it makes you think about how likely it really is.
And Dr. Tour is pretty straight up about how he thinks these gaps in our understanding are often filled. He suggests that sometimes when it comes to explaining these big evolutionary leaps, storytelling takes over.
Storytelling. That's a bold statement. Is he saying that some evolutionary explanations are more like stories than actual science?
He's definitely challenging the scientific community to give more solid evidence for those big claims about evolution, and this brings him to a bigger point about honesty and humility in science. He says scientists have the responsibility to be clear about what's a fact, what's a theory, and what's just an idea.
That's a good reminder. It's easy to get carried away with a new discovery and exaggerate its importance.
And he's worried that presenting speculation as fact will make people lose trust in science.
Yeah, that makes sense. People need to feel like they can trust the scientific process, and that takes honesty and transparency.
Exactly. And he's especially critical of people who shut down anyone who questions the accepted scientific theories, especially people who have religious beliefs.
It does seem like there's this assumption that science and faith just don't mix. But Dr. Tour seems to be living proof that you can be a serious scientist and still have faith.
Absolutely. He says that open and honest debate, even about tough topics, is super important for science to move forward. Shutting down different viewpoints just holds back knowledge.
This has been a really thought-provoking deep dive. We've gone from the mind-blowing complexity of cells to the challenges of explaining where life came from and the limits of what we know about evolution. So what does it all mean? Where do we go from here?
That's the question Dr. Tour leaves us with. He doesn't give us easy answers or say he knows everything, but he wants us to think critically, to ask hard questions, and to be open to different ideas.
It's a powerful message. It reminds us that science is a journey, not a destination. And the most important thing is to keep asking questions and keep exploring.
Absolutely. And if Dr. Tour's lecture taught us anything, it's that the more we learn about the universe, the more we realize how much we still don't know. There's a sense of wonder and humility that comes with tackling these big questions.
Totally. There are so many mysteries out there still waiting to be uncovered, so many questions waiting to be asked. So keep exploring, keep questioning, and keep diving deep.
And on that note, that wraps up this deep dive. Until next time, keep those brains buzzing